Yancy and NSTF, thank you for inviting me to participate this afternoon. I've been listening to some of the talks even yesterday quite keenly, and it's very informative, so thank you. This afternoon, I have been asked to talk about some challenges in research development and innovation where biophotonics is concerned, and that's the field I also work in. So I would like to just, take the audience through a few of the opportunities first, and then I will mention some of the challenges, and then I also would like to take an opportunity to touch on some of the innovations that my team specifically works on, just to display that we can be a contributor in the biophotonics markets, not only us, but the researchers that are working in biophotonics in South Africa can be contributors in the markets because there is an opportunity, and also we can use our research to also maybe improve on the on our GDP, our country's GDP. So because I'm not sure who is in the audience, I think naturally it just also occurred to me that maybe I must just mention what biophotonics is. So biophotonics is really a science of harnessing photonics or using photonics to understand the inner workings of living cells as well as tissue. And this is the case when you are basing your assessment or analysis on the medical side of the research. Biophotonics can be applied in a number of fields, including the beauty industry, the pharmaceutical industry, as well as the agricultural industry, to mention but a few. So we are focusing on particularly the medical industry as well as the pharmaceutical space. And that's basically what biophotonics is. And when it comes to the markets, there's great opportunities in the biophotonics space. You can see that the market is anticipated to grow from about 53 billion US dollars in 2024 to about 141 billion US dollars by 2030 at a compound annual growth rate of about 11.3 during this focus period. So definitely there's huge opportunities in this space. Zooming into the markets and just looking at the contributions via region, regionally, let me just get my laser pointer. The biggest contributors of the pie are North America, followed by Asia. And, I don't think it's a picture that's different when you look at markets in different industries, across industries. This is the picture you will pick up. When it comes to the contributions from Africa, and the Middle East, this is where we lie. And you can see that really it's not as efficient. So we need to change the picture somehow. And I was also keenly looking at the key players, and you can see all the companies that are, the key players in the markets, but also the end user segment overview took my interest. You can see how it's growing from 2022 and how it's focused to keep growing by 2029. And medical diagnostics lead that side of things, followed by medical therapeutics, tests and components follow next, and then finally it's all the non-medical applications. So a few years ago, we had an analysis of how much we are contributing to the Photonics Centre, not only the biophotonics, but the photonics sector in South Africa. And you can see that the market share was less than 1% at the time. And this was in 2013 when the photonics markets, like the entire photonics markets, was around 510 billion US dollars. The picture hasn't changed much. I mean, I haven't done the recent calculations and analysis to see where we sit in 2024, but the picture has not changed much. And some of the challenges really when I look particularly in the RDI space in biophotonics, where I'm sitting with my team, include the likes of your skills training, like capacity building skills training, and human capital development along that field is very difficult, because when you get resources, they come with, plain physics, plain virology, plain mathematics. It's very difficult to get highly skilled individuals joining and working in the field. So this is one of the biggest challenges. You basically have to train your own, so you have to grow your own timber. And that also comes with its own challenges, because sometimes, people get better opportunities when they are highly trained and they leave. And, it becomes a bigger challenge in that sense. And then infrastructure is another big challenge. And these are not really unique to the field of biophotonics. These challenges are not unique to us alone. I listened yesterday, and a lot of other, talks were talking towards these challenges. Infrastructure, particularly when it comes to labs that allow multidisciplinary work to happen in the same space, is a very difficult one as well. Not necessarily maybe for us, because we have managed to set up and, are fully set up. But I've seen in some of the labs that do biophotonics in the country, and I collaborate with, that infrastructure is a challenge. Something else that is a big problem is that of translating research from the bench top to getting your products, commercializing your research. And I listened quite keenly to the NIPMO presentation yesterday. And yeah, this area is, of the, like for us at the moment, because we are tech developers, is very important, but is also very frustrating, because it's very difficult to get your research from the benchtop to the markets. The issue of collaboration is another one that's a challenge, because culturally we work in silos, and it becomes very difficult to then, learn and grow from each other, because we are just scattered. So collaboration is actually quite key as well, and it is required. Bureaucracy is another one that is a big challenge. And I'm not anti-structure, but sometimes, we, for example, get most of our equipment as well as components from overseas. And procurement becomes really one of the biggest challenges, and that's because of the bureaucracy behind our procurement protocols. And attached to that is regulatory requirements. Lab regulatory requirements also have got, they're quite bureaucracy intense. And as I said, I'm not against structure, but sometimes we incur severe delays trying to deal with the bureaucracy that surrounds some of our processes. And of course, funding is also a challenge. So, because I don't want to spend all the time that I've been given so graciously by NSTF to share this talk and not go into the research that we do. And the reason I'm also doing this is also to show that biophotonics can be a player that can eventually, as I said earlier, even contribute to our GDP in the country. And of course, I'm going to be sharing some of the work that we do at CSIR, but it's not only CSIR that does biophotonics research in the country. There's several other institutions that have biophotonics research. So at CSIR, we are working in the space of diagnostics, and we've branded the work, or we are working towards the development of Ukukhanya Diagnostics. And Ukukhanya is a Zulu word for light. When you use it as a verb, it means to shine. Used as a noun, it means brightness. Why light? Because we are using light, be it lasers, LEDs, or any other type of a light source. And we are taking that light source, we are shining it onto molecules. It could be your biological molecule, could be your chemical molecule, to be able to tell you what the composition of the molecule is. And this allows us to do diagnostics when it comes to communicable diseases, non-communicable diseases, as well as substandard medication. We are in a space that's multidisciplinary, and because of this, our projects cut across the photonics, which is the heart of the research, really, lasers, your optics, everything photonics. And it goes on to analyzing pathogens. So we've got your virologists that understand how pathogens work and can inform the research along that line. But we are eventually wanting to also have a footprint in the space of medical device development. And this is a unique feature that is quite exciting and makes working in this space, quite critical. So let me talk on some of the technologies that we have come up with to date, and that show really like huge promise when it comes to product commercialization. One of such products is a smartphone diagnostic that we have designed. And the problem statement there was that there is poor access to healthcare in our country. For people that live in the cities, it's very easy to go to the doctor, who will then tell you to go to write you a letter to go to a diagnostic lab, be it a public or private lab, to go have your bloods drawn, and then they are then tested, and then you get your results in a few days. For some of our people in South Africa that reside in rural areas and as well as peri-urban areas, just like this group here that's based in KwaZulu-Natal, they have to rely on a mobile van that comes, I don't know the frequency, but it comes ever so often to collect the bloods, then transports the blood to wherever in the labs, it gets tested. So, it's longer to have diagnostics or tests taken in such an environment. And with disease progression, we know that is something that needs to be dealt with very quickly to be able to give the patient treatment that will work for them as quickly as possible, so that we circumvent disease progression and worsening of symptoms. So because of this, what my team and I sat and thought was a possibility of putting a laboratory like that in a smartphone. So with our optics and photonics, we were allowed by the technologies to create both software and hardware that is novel and inbuilt in a phone. And the idea really is to test any bodily fluid to be able to inform a patient what is it that they are suffering from. So it's all also inbuilt with artificial intelligence as well as machine learning in order to, do all the analysis. So the technology is at technology readiness level 7 at the moment. We are validating. We are also going to be using infectious samples to finalize the tests on the mobile phone, and then we'll be able to further commercialize it. But attached to the smartphone, which I've just been showing now, smartphone biosensing, we have other optical systems that we have built in the lab. And this slide really is to just show some of the work that we do in our space. And if there's maybe members of the audience that are listening out and would like to collaborate with us, these are some of the technologies that you can expect to find in our lab. So we've built a localized surface plasmon resonance system that is used towards diagnostics of pathogens as well as non-pathogens. There's a photonics crystal biosensing setup that we have put up, and we also have a enhanced Raman spectroscopy setup. And here I'm actually showing some of our results that we have published, showing HIV infected cells over a number of days. I think the orange trace was day 2, day 4, and day 6, the purple trace. Our space also allows us to run some of the gold standard tests like the ELISAs, the PCRs, but we also have a fully fleshed cell culture laboratory, and we are able to prepare different sample types. Another technology that I think would be worth sharing this afternoon is that of the loop-mediated isothermal amplification. And for the non-technical people, please excuse me. The idea is not to, overload you with jargon, but just to show some of the things that we do in the lab while trying to keep it simple, really. So the idea here was to actually simplify what a polymerase chain reaction machine can do. This is a PCR machine. I think the audience would be familiar with this. PCR tests are done for, when during COVID, when the samples were taken, they were tested via PCR. HIV presence is also tested via PCR. So does TB, and the test, it's a gold standard, it works, but it's tedious. It's a tedious setup to operate. It's time consuming, it's high equipment costs and maintenance, and we then looked at how we can mimic this using artificial intelligence and also putting the platform on an Arduino-based platform. So we've come up with a handheld Arduino platform, and that's the camera right there. You can see it on the screen. It's pointing on the keyboard of this laptop, and these are the electronics. The system doesn't look like this anymore, and I'm just going to hide that for now because it's also under IP protection. So the idea here for this innovation is to simplify and make the current technologies very simple. We would like to come up with this LAMP Arduino-based approach to simplify the processes, reduce the dependence on specialized equipment, and also to improve accessibility in molecular diagnostics. The other one that I would like to touch on this afternoon is that one of substandard drugs in Africa and how we are addressing this issue. So you would see the report from the World Health Organization that 1 in 10 medical products in the developing countries is a substandard or falsified product. And this is a problem because people are consuming medications, but they are not getting better. It's not that big an issue in South Africa, while it is like a problem, but in other African countries, it's a nightmare. So to try and solve this, what we did was come up with an optical means where we build novel chemical sensors to actually pick up the different molecules in medicines. So the gold standard include the likes of your HPLC and mass spectroscopy, which are highly sensitive, but they are laborious, expensive, they are high maintenance, and they also have longer turnaround times for results. In our case, we're using surface enhanced Raman spectroscopy with novel sensors, and it's coming with ease of use. It can be digitized, it's low maintenance, and you get your results within minutes of running the test, and it's compatible with 4IR as well as machine learning. This is one of the data sets that we got after we actually tested antiretroviral medication. We screened it, and this was the pure sample of the ARV, and we spiked it. And you can see that there's definitely a difference in the two traces when you are using pure versus spiked. And this is one of the technologies that we would like to miniaturize so that we can also be able to take it wherever where the need is to screen for medicines. Another application of our work, something that we feel is a big opportunity as well, is that on analyzing indigenous knowledge simple systems. So, samples like this where you have a whole load of traditional medicines. Sometimes with traditional medicines, it's not really known what the composition is. Some of it is still being classified. So we can use our optics tests to actually classify medicines. We've already started doing it with some of the traditional healers that we work with, as well as one of our groups here at the CSIR that's doing IKS have sent us samples to try and classify and to be able to inform them what those samples compose or rather consist of. So that's another huge market right there. Attached to this really is also markets in the agricultural space, which we haven't started working in that, but that's where we are going, as well as in the beauty industry. So when it comes to opportunities, really, they are plenty. And as part of my conclusion, I would like to say we've seen that reports show that biophotonics markets are to increase to about 141 billion US dollars by 2030. And I don't think that we should let this opportunity slip our fingers, because really, as I've shown you now, technologies are there. It's possible, it's very possible to train human capital that is highly skilled and that can work with these technologies. And you can make the products. I mean, we have made products. Yes, we are incurring delays in terms of getting them off from the bench top to the markets, but it's possible to do all of that. So we can really capitalize on this opportunity. And one of the other things that I would like to mention as maybe a potential solution to some of the challenges that I mentioned earlier is to, come up with a culture of diversifying work in South Africa, whether it's in science councils or in university. Let us support multidisciplinary work. Silos are not doing us any good. Also branding ourselves as physicists or biochemists or molecular biologists or virologists. I think the days for that have come to if they have not, they are coming to an end, because current research evolution requires that we join the disciplines and we feed off of each other. And then attached to supporting multidisciplinary research, it's very important to also develop a strong pipeline, even, going all the way down to high scholars, where high scholars are exposed to fields like photonics, so that, they grow with that type of exposure and knowledge and awareness. And, the idea there would be to capture them while they are still young, show them the importance of the field, and show them what can be achieved using photonics research, and just develop a strong pipeline that is aware of what is out there where photonics is based, and with the hope that they will then take it to postdoctoral level, their photonics training, and that will be the next generation. So strong pipeline, very important. Another thing that I thought would be worth mentioning maybe in my conclusion is that of research translation. And as I said, it's a big problem, and it delays so much of our keenness in commercializing technologies. So here really we need to have an integration of ideas from different stakeholders. And I don't only mean people that are working in photonics, but I'm thinking, healthcare producers, the manufacturers, the regulators, policymakers of sorts, suppliers. We just need to get to a point where we are communicating. This will also lessen the bureaucracy that's surrounding some of our processes, because if everybody is singing from the same if we are all just taking the same approach towards solving a similar problem, we might come up with ways of also making sure that when it comes to processes, it's easier to attain some of our objectives that we would like to, at the end of the day, realize. And then also to enhance collaboration, it's very important that we work together in the sciences and within universities as well as science councils. What I've also seen work quite well in my space is that sometimes when we have a problem that's like recurring and it's not, it doesn't seem like we are getting a solution quicker, sometimes disrupting the KPIs as well as the KRAs, key performance indicators, as well as the key result areas it's surprising how much that can do, you know, just change things around and make it a requirement. And if something is in my KPI or KRA, I know that, you know, it doesn't matter what, I have to work towards obtaining it. So, and you know, I'm just using this as an example of what works in my space, but I'm sure it can work across the spaces. Also, we are also working towards diversifying funding streams, because, you know, just applying for competitive funding or applying for government funding, whatever the case may be, yes, is very helpful. And we are very grateful to those that have supported us to date, but we have to find ways of diversifying our income streams, and that's what we are working towards. So, I think that's where I will stop for this afternoon. Because I mentioned some of the work that we do, I would also like to acknowledge my team. These are my team of researchers, and these are the people behind the work that we do. They are standing in one of our molecular biology laboratories, and thank you.
Right, Busisiwe Lant says, thank you for a very informative presentation, Patience. Kindly advise on the following. To classify or screen for API or novel molecules in medicinal plants, in what form do you require the samples? Can traditional healers send their samples directly to you, or should they go via the CSIR?
Okay, thank you so much for that question. So let me just go on video. Oops, sorry. So we can test you know, samples in different states, liquid state, if you give us if we have a sample that needs to be, you know, if we grind it as a solid state, we can grind it and then also get it to, we can take samples that are solid state, but sometimes if we see that the spectra are compromised, then we get it to liquid state and concentrate it. So, you know, we can test different states depending on the sample, and also depending on how we can influence or change the different molecular composition of the sample. We don't want to tamper with the sample too much. So we try and, you know, create technologies that allow us, for example, when it comes to doing screening of medical devices, we want to get to a point where there is as little sample preparation as possible. You know, you just want to test whether something is in a box or it's, you know, it's in a container. You can still be able to tell what the content of the box or the container is. Okay, so that's where we want to push the technology to. But at this stage, liquid state, solid state, it works. When it comes to the question of how do we get the samples from the different partners that give us samples, we once again have to face up with the bureaucracy. There's ethical clearance, because everything is governed by policies, and you know, you can't just take a sample from the street and bring it to the lab. You know, it has to follow a particular protocol where you eventually can then test it in the lab. So yes, we follow regulatory processes that get us to a point where we can test samples in the lab. For the colleagues that work with us at the CSIR, and recently the colleagues that work with us from TUT, the Cannabis Hub, we can get samples directly from them because they already have MOUs and the necessary documents that have been you know, agreed with the CSIR. So yeah, there's regulatory processes that have to be adhered to. But yeah, once those are all done with, we can be able to test the samples.
Okay, thank you very much for that answer. Are there any other questions for Dr. Mthunzi? I don't see anything else in the chat. Dr. Esper.
Afternoon, and thank you, Prof, for your presentation. I'm wondering because I'm listening. You are talking about established institutions that you've already have got MOUs. Let's say I'm an indigenous person situated in Limpopo. I've discovered that this particular traditional plant can cure cancer or a particular disease. And I want you to do this confirmation. How do I go? I don't know any regulatory process.
Okay, so if you start conversation with us, we can get the necessary people, like we can get our IP office in place, we can get advice from policymakers that can tell us how to go about it. I don't think that it's impossible to work with you know, people that are outside of the big organizations, but all I'm saying is there are processes to be followed. So it's not impossible, but it's not just a matter of also getting your sample to the lab, you know, without having it registered or all of that, because there could be issues with IP. Maybe your sample has got, you know, potential to be patented or whatever the case. We need to do right by you. So if we don't follow the necessary processes, you know, that might not be adhered to. So yeah, there is a way around it. So if you are interested, I think these talks are being recorded. You can send me an email and then we can have a meeting, sit down with our IP office and the other policymakers and see how we can then get the work with you or with testing your samples.
Yes, I can see your slides.
Thank you very much, Yancy. And thank you for this wonderful opportunity. I see a hand up from Dr. Espany. I don't know if you'd like to attend to them first.
No, that is a previous hand.
Oh, okay.
You may go ahead.
Sorry, it was a legacy hand.
Thank you.
Thank you. And I would like to thank everyone for actually attending this session and for still being here. Can you see my slides, Ansi?
Yes, I can see it clearly.
Can you see them clearly? Okay. So my name is Babaki Malopi-Khokong, and I'm the National Manager for Academic Affairs and Research. And I'll be talking on the RDI challenges in the public health sector with focus on the National Health Laboratory service. So just as a background, the NHLS function is to provide cost-effective and efficient health laboratory service to support health research and training. So we have a triple mandate, which is diagnostic service, which is the core. We also train for health science, mainly pathology and public health and occupational health. And we also are supposed to be supporting research. We were established in 2000, in year 2000, through the National Health Laboratory Service Act. We are the sole provider of diagnostic service in the country. So we therefore serve 80% of the South African population. So it will be 80% of the 60 million plus South African population. We employ 8,000 staff, and we have laboratories in all 9 South African provinces. And we are very important to the South African healthcare system because of our role as the sole provider. We also just want to make everyone aware that in addition, NHLS has subsidiaries or institutes. The NICD, which most people may be familiar with as part of the NHLS. The National Institute for Occupational Health is also part of the NHLS. The Forensic Chemistry Laboratories, which were recently acquired as part of the NHLS. We also have the South African Vaccine Products, which we are the only Southern African producer of equine antivenoms for snake bites, spider, and scorpion stings. And we also have the Media Diagnostic Product, which is part of the NHLS. Just an overview of how big the NHLS is, as indicated. You find us nationally, everywhere in the country. We are in 422 public hospitals. We actually service 3,841 public clinics, and overall we have 233 diagnostic and clinical laboratories across the country. And we are in all the 9 provinces, and we're serving about 16 million South Africans. And our values as the NHLS, I think what's important to this topic, we strive for transformation, we strive for service excellence, we also want to lead in innovation and continuous improvement. Obviously, we want to be in a situation where we provide excellent diagnostic intervention, and also we are improving the clinical interventions for all the public health sector patients. Also, what is important is that as the NHLS, we are actually in partnership with the 11 South African medical universities. So for the schools of pathology within these medical universities, we employ the head of departments, we employ pathologists and medical scientists who are joint appointed staff between the university and the NHLS. And therefore, our staff are expected to provide service. They're expected to teach and train. and they're also expected to conduct research. And then in addition, we also have formal partnership with the 6 universities of technology and 2 comprehensive universities, which will be the University of Johannesburg and the Nelson Mandela University. So what are our key concerns when it comes to RD&I? I think I've already indicated that our staff, they actually have to provide diagnostic laboratory service. They actually have to do teaching and training. And this they have to do because we have to make sure that we provide the service to the patients and the clinicians. We have to make sure that our trainees are taught. And then our third mandate, which is research development and innovation, is therefore compromised. It's not prioritized. And also as the institution, it's not really incentivized. So it's not really attractive to the researchers. And also our leadership, obviously, they sometimes are bound to make decisions between laboratory service, teaching and training, and research development and innovation. And you find that research is actually, I always, being the head of academic affairs and research, I always find that research is the third priority amongst the 3. And therefore, we actually need top-down approach and clear goals and direction on research, because it's clear what everyone is expected to do on the service platform and the training platform. But when it comes to research, we need to make sure that the direction is also as clear and the expectation is also as vivid to everyone. We need a clear innovation strategy, which is linked. I also had the previous speaker talking about key performance indicators for research and innovation. Because if these are not properly linked, then it also is not priority or it's not going to be prioritized. We also need innovation dedicated trainings and inductions. I think we still come from a culture of conducting research, but actually we're still trying to understand what is innovation, and especially because it's not incentivized, is it really important to do that? And also, how do I move from my research mode to the innovation and technology transfer mode? We also need active engagement to encourage inclusive innovation think tanks and follow-ups and policy adaptations. And what is important, I also heard it was mentioned, is flexible and diverse funding approaches. And this is not only talking to us as an institution, but also it's talking to funders out there. Because currently we know that research is really funding, it's specialized, and which actually restricts creativity, and it's very specific to the outcome. So what are the current general challenges that we see? I thought we've spoken about lots of challenges on innovation holistically. And one of the key things I wanted to zoom into the institution, and which I think is also a challenge with the public sector, is inflexible HR and procurement processes. Especially with the NHLS, we have service, we have teaching and training as core. And now our policies are focusing on the service platform, and they're really not flexible to research and innovation. So what happens is that we have to follow the standard PFNA, we have to follow the standard HR policies and practices. And one of the key things is that you find that when it comes to research, which it has to happen now, commitments on procurement and recruitment of staff and turnaround time are very uncertain. So even when you put an application, you never really know if you will have a staff member in the next 2 months or in the next 3 months. We have a misconception in deviation because we are solely focused most of the time on procurement for service. Procurement for research and innovation is not really understood. So what happens is that if you have a specific project which requires product A, procurement is still wanting you to get quotations for product A and product B and product C so that you can meet the PFMA requirements. So we find that lots of sole supplier deviations have to be done by researchers, which actually leads to delays in procurement. We also find that what we think is supposed to be an urgent procurement or HR request, it's actually not being prioritized because it's service first and then everything else comes later. And then we also find that there is communication breakdown because most of those who are providing service to us, they are focused on the lab diagnostics and they are not understanding the research side of things. And you find that whatever you say may not be understood, So while we see the 6, they will be looking at the 9, and this is very confusing to us. So also with HR, we know that with research, we actually have short funding opportunities. So there is increased skilled talent turnover. This can be controlled by proper planning, by policies, but our policy are not flexible enough for you to circulate the stuff which you're already having. If the contract ends and you're still waiting for additional funding, then the contract will be terminated. So it means you actually will have a talent void because what happens is that you are continuously on a rotating recruitment phase where everyone who is there, it's been circulated and you get new staff almost every 12 months, depending on the project. And also it means you are attracting less experienced talent because people don't want experienced people don't want to be in short contracts, and they also don't want to be, to always be looking whether to worry about whether their contracts will be extended or not. And also, I think this also leads to skill transfer limitation because what happens is you train somebody this year for 3 months of probation, they work for 9 months, and then you have to train somebody else. So what are we trying to do? We are trying to communicate with our support departments and to say, there are clear facts about funding, especially grant funding and RDI. PFMA and HR policies, which we have to follow strictly, are not research and innovation friendly. And why? Because, for example, grants funding is usually provided for short periods, which, in most, it will be 12 months, it will be 36 months, and if you are lucky, it will be 60 months. And we have to make sure that during that period, the principal investigators are pressurized to spend the funds and to complete their projects within the awarded period. But also, our policies do not allow for you to initiate maybe HR and procurement processes before funds are approved and granted. So because of that, obviously, it means you will have delayed procurement and recruitment processes, which leads to projects not completed timely and project targets not met. Requests for award extension or carryovers that has to go to grantors. They are also dependent on grantor turnaround times, and also it depends on grantor regulations. Some may approve the extensions and some may not approve the carryover of funds. Grantor refunds, if the funds are received in advance, you may have to be returning those funds, and some grantors will also say we want the end interest to be returned. And also organizational reputation. So if our policies are not flexible, if our PFMA, we don't see how we can align appropriately with it to fit RDI, then obviously in some of the research projects, we are not meeting the objective on time. We always have this reputation of late objectives being met. And obviously, organizational reputation is affected. And also, we are seeing declining in PI confidence. So because we have most of our staff who are conducting research as joint appointed staff, then most of them, if they can, will move the funds to the university because they have better turnaround time and they also have flexible policies. So some of the interventions which I think we have to actually, from the planning of our research, talking to the researchers, talking to the grantors, talking to the research support officers, and actually talking to leadership, is that we need proper planning and scheduling. And what is important is we realize that we focus on submitting the proposal, submitting the grant application, but then you'll find that even when targets are set and timelines, they are not really clear. And also we are not considering setbacks. So even if we know that we may not hire somebody in 3 months, we are not saying, what am I going to do during this period? We are planning as if it will happen as it's supposed to be. But also we need to make sure that we have extensive progress tracking, because during that period, as you track progress, as you note that there are some things that don't work the same way, we have to be immediately reactive. And then obviously supply chain management, we need to build relationships. We need to make sure that we communicate effectively with them. We need to make sure that they understand the reason why they have to be flexible. But what is important, they need to be very responsive, and they need to understand why, especially in our scenario where they think that because you are research, we have to prioritize the patients. And then what is important also is contingency plan, just in case scenario. So if this does not go this way, what is my plan B? And this is one of the things that researchers have to do and innovators have to do from the beginning. Because if you don't include the contingency in your application, our funding is not flexible enough to actually accommodate that contingency. And we have to make sure that we think beyond what we think is normal. I think we all know of the current NHLS situation. We've been cyber attacked, and because of that, it affected our service, but obviously it also affected research. And because there are several students, there are several organizations who are depending on us to supply some of the materials, some of the data for them to conduct research. And one of the things that we have to learn is risk management and risk mitigation. If this happens, what am I going to do if something goes wrong? And in this, I think I'm in a situation where we are at, it went wrong, then what? And you have to make sure that there's a clear plan. And this is what everyone has to be taking in consideration. And the next thing is the institution having now being aware of the situation. What are we doing? As the NHLS, we know that we are supposed to support research as one of our mandate. So one of the things that we are doing, in training and development, we are actually training recently at least 50 medical scientists, and we recruit new 50 medical scientists in 10 every year, and we're recruiting at least more than 60 pathology registrars every year. But also what we do, we also have a virtual training platform, ECHO, and we have set platforms across the country, and there's 93 sites nationally across the NHLS laboratories. And through this, we are offering CPD accredited sessions annually, and we've been offering more than 230 sessions in pathology, in diagnostics, in research, in public health and occupational health annually. We have trained more than 2,000 people since 2018, and we also have dedicated RDI sessions. As an NHLS also, we are making resource accessible for researchers. Whether you are an NHLS staff member or you are external, you can request access to de-identified leftover samples for your research. You can request access for anonymized data for research, which is where I'm saying most, you'll find that there are students and researchers who are affected by our current system down. We also are encouraging collaborative opportunities with NHLS experts. And what happens is you can come through our office and we will provide direct links to you. And we will find the direct links across all the universities that we have partnership with. And also applications can be submitted on our ARMS portal, which is accessible externally. I could not provide the address for now because our ARMS portal is currently not active. So we are also very open to partnerships. We obviously are working in university multi and inter collaborations, but we also work with Science Council, the CSIR, HSRC, SAFC, and many more. We also work with NGOs, Caprisa, Right to Care, Basel Consortium, and we also work with international collaborators. So this is just the research material and data access that we get annually. We introduced our ARMS system around 2019. And since then, we've been receiving on average about 300 applications, either to access samples or leftover samples or to access our data for research, and all the applications are done through the ARMS portal. And in terms of our research output, we are publishing about, in average, more than 600 publications. And for this year, our publication output was a little bit low. We can see that 2021, we had a peak. We're thinking this, actually not thinking, these were mainly COVID publications, and we saw the decline in COVID publications, but we were hoping that we will actually peak for this year. And we are still trying to understand why we have this decline, but we are thinking it may have been the COVID effect, but also we are thinking it may be the innovation effect, because we have been trying to promote innovation with our researchers and asking them to take their time before they publish. We know that most of our old researchers are not happy with that because they want immediate, satisfaction of I have published, instead of waiting years without knowing what's going to happen. So overall, we had more than 3,000 journal publications in 5 years, but our innovation drive is still low. We had 2 innovation disclosures, and we had 2 patents in collaboration with UKZN, and the second one was in collaboration with Caprisa and 2 USA institutions. And in addition, we also have a PathReg Congress, which is held every 2 years. It's mainly pathology and public health research. And in the last 2023 Congress, we had more than 1,000 actually attendees and registered people. So we had about 97% attendance, and we had more than 312 submitted abstracts, and majority of those abstracts were from NHLS researchers and NHLS collaborators. So what does the NHLS do to try and drive RDI within the institutions? So one is providing funding opportunities. And one of the things is that we have negotiated with the NHLS Research Trust, and they have approved a pathology doctoral support fellowship, which is 1.5 million and will be introduced by end of this year. So this is to offer pathology time off to go and do research. And then also we have multidisciplinary pathology collaborative program, which involves 3 institutions, and one of those institutions have to be an HDI, and this will also be introduced by end of this year. And we also have a few funding opportunities to just kickstart the young and upcoming researchers within the NHLS and encourage them to understand and be able to submit to kickstart their own grant application processes. And also, we are looking into improving research development support. And what have we done is now, from this year, we actually hired grant-specific procurement staff. So we have 3 staff members who are only focusing on grant supply chain management and making sure that they are trying to actually make procurement understand the researchers' needs. And we are seeing this being very effective. We've also placed a grant-specific human resource staff who is only focusing on grant recruitments and not competing with the overall NHLS service and training recruitments. And we're strengthening our grants program unit and our grants finance unit. And through the Need More, we are thankful that we are actually strengthening our technology transfer. And also, in terms of funding, our grants management unit is actually managing funds between 700,000 to 1 billion annually. But of concern is that 24% of this is international funding, 25% is local funding, and only 1% is from the institution. And obviously, if you want to drive specific objectives or specific goals on research, then we really need to strengthen our local funding and our institutional funding, because in this case, we are driven by what the grant hosts want you to do. And within the NHLS also, we have bodies. We have a research development committee of the board, which is only making sure that the research mandate of the NHLS is delivered. And we have the strategic research and innovation plan, and we have policies for RDI, and the specific unit academic affairs and research, which is now focusing on research and development. So in conclusion, from my side, I think we have spoken a lot about innovation and the setbacks. But I think what is important is that we need the flexible funding opportunities. These have to be diverse. We need grantor to understand if we're to innovate. Sometimes ideas changes halfway. We need innovation skills to be developed, but also we need buy-in. We need researchers to know that they have to be focusing on innovation than focusing on publication. And this is our case as the NHLS. We actually need open minds of everyone here to multidisciplinary and multi-institutional partnerships. I've indicated we do have partners with NGOs and Science Council, but negotiations takes long because we see non-flexibility when it comes to IP sharing, because everyone wants it to be mine. But if we want to be in partnerships, then the idea of IP sharing has to be clear and has to be transparent. And also, we need to make sure that we have effective collaboration. And as the NHLS, we actually are calling towards any collaborators who are working towards the enhancement of laboratory diagnostic, clinical intervention, and service, laboratory service efficiencies. We are happy to welcome you and to work with you. And we're just hoping that whoever comes, it will not take us 6 to 12 months to actually conclude a contract. And what is important for the NHLS is institutional rethinking on how to drive innovation and to promote research translation. Thank you very much, Yancy.
Wow, Dr. Kokong. I was reminded of how vast the NHLS is and the enormous mandate that you bear. And I want to wish you all the best with the shift towards increasing RDI and strengthening that aspect of your mandate. Thank you very much indeed for this presentation.
Thank you.
And for attending the entire day. I really appreciate your presence here and your interaction.
Thank you. Yeah, it's actually the entire 2 days.
Yes, the entire 2 days, I should have said. Yes, indeed. Thank you very much indeed. I'm going to move on to the last speaker, but I'd appreciate it if you could stay on, Bhavati, for the discussion, the questions afterwards, after her presentation. Would you be able to?
Thank you. I will.
Thank you. Our last but not least speaker for the day is Professor Melanie Lake. She's president of the South African Education Research Association, and she's talking about CSIR, supporting young minds, strengths and challenges. Welcome, Prof. Lake.
Thank you. Thank you very much. I hope you're able to see my presentation.
Yes, able to see it. It's not on presentation mode yet.
I think it is now.
Yes, there it is.
Thank you. Well, thank you, everybody. Thanks for this invitation, and thank you for allowing CSIR to share our perspectives around RDI and the key interventions and activities that we engaged in order to address the issue. So today, the focus of my presentation will just be a very brief outline of who and what CSIR is and what we do. One of the key RDIs and issues around this RDI that we want to focus on is the challenges of mentoring young scholars and researchers. And I specifically want to look in the presentation today at how CSIR addresses these challenges, and at the end come out with what are the key strengths of the activities of the organization to support this very important challenge. Just a little bit about CSIR. The aim of CSIR is development, and it basically is to enhance the education as a research field, and to develop capacity around the scholars and the country's educational development and progress. So we play a very, very important role in nurturing and supporting this development. Within our aims, our two key aims is to grow the organization, and the organization has grown over the past more than 10 years into a leading organization where many researchers, many people outside of the research arena value CSIR as an organization in order to network and develop. And we're particularly interested in supporting capacity and the development of education researchers, but one of our key goals is to mentor and grow young researchers and leaders, research leaders within South Africa. But I want to be upfront about what the issues and challenges are that we experience. So I just want to give you a little bit of an idea around the makeup of the organization of CSIR. So in CSIR, we have an executive and we have members. The whole executive consists of individuals who are actually researchers active within institutions, higher education institutions in South Africa. So every 2 years, a new group of executives, we develop our executives over a period of 2 years. I'm currently the president for the next 2 years, and I'm hosted at the University of the Western Cape. So our executive consists of people who are active in research, who are working in institutions, and we ourselves take the key challenges as something coming from the institutions that we're working in with our memberships, our members who are also researchers from various institutions within South Africa, and also some organizations within South Africa make up our members. And so we are working with people on the ground, and we are actually working in the institutions. And these are the challenges that CSIR picked up, and the important notes about developing young researchers and scholars in the field. One of the important, the first would be the uneven, it's the issue around mentorship. Mentorship, particularly at institutional level. Remember, we work across many higher education institutions. And within these institutions, there's an uneven level of institutional mentoring. We also have our particularly young scholars and developing scholars having limited access to established and influential scholars, and partly because the more influential scholars, there's very limited time to engage. And while they do engage in organizations like CSIR, in our organization, very actively, sometimes there is limited access to these influential scholars. For instance, A-rated scientists, we could also say chairs, NRF chairs, etc. There's also very little scope for emerging scholars to interact and learn and share. And as I will talk through the presentation, we would show you as CSIR how we give these scholars opportunities to access each other and also to network. And at the margins of research innovation, so many young scholars find that they're always on the outside looking in, and sometimes those opportunities need to be made available to them. And that's what we try to do through our organization and give opportunities for this kind of engagement. And there's limited national and international collaborations, particularly for younger scholars, and they would need access. So CSIR as a national research organization has made strides in addressing some of these challenges. And the way I'm going to address it as we go through the presentation is by looking at the activities of CSIR in relation to some of these issues that we've addressed now. So how we address some of these issues is within the key pillars of CSIR. So CSIR is an organization made up of many activities, and the activities, one of our biggest draw cards is our annual conference. The conference is hosted by different universities every year. We also have people development and networking as an umbrella. And here we have our members. We have people development and leadership through many activities within the organization. Communication and networking. So previously we spoke about scientific, one of the presenters spoke about scientific communication, and that is key, that scientific engagement and communication with the community plays a very important role in our organization. And one of our key ways of disseminating knowledge, besides workshops, the conference, etc., is our Journal of Education. Our Journal of Education also allows exposure to cutting-edge research, but also our conference and the presentations related to our conferences allows this access to cutting-edge research. So CSIR as an organization, we are extremely influential in addressing some of these challenges. And while these three components play an important role, we do have to note that they do overlap. There are a lot of overlap. And what we find is this overlapping provides a rich environment for our young scholars to engage. So in red, I've highlighted the issues that I spoke about previously, the challenges, and I want to tell you within our strategic objectives and priority how we address and what activities we offer. So we are providing, having identified these challenges, we are providing a space to open up opportunities for our young researchers, young minds, and the growing researchers in our organization. We have the annual conference. It's hosted by an institution, and we organize this that there's a 3-year. We know for 3 years who the next university would be to host the conference. The conference is while we do support the LOC, the Local Organising Committee, the conference is organised by the LOC within that institution. What we do encourage is that people within that the university get involved, and here we have many young researchers actively involved with CSIR and developing the conference from the beginning of the year to the annual conference, normally taking place at the end of October. So this year, our annual conference will be taking place at the University of the Free State from the 29th of October to the 1st of November. And so we have had our submissions of abstracts already. The closing date was in July, but you're very welcome to join our conference just to listen to the presentations and to see how the organization engages. Within the conference, we also target themes, and we allow the LOC to target themes that are current and research-driven themes where participants are able to get involved through their cutting-edge scholarship and presenting it at CSIR. And most commonly, many education researchers do come to our CSIR conference, and we often have very cutting-edge and updated presentations presented within our conference. Within the area of our people development, so one of our key strategic objectives is to develop our membership, leadership, and networking. But we need to provide those spaces. So we do recruit members from all universities in South Africa, and you're able to be part of CSIR. There are also outside organizations that are part of the CSIR membership. We open spaces for early career researchers to support, grow, and expose them to our organization. So while there is very limited scope for emerging researchers to interact, that are one of our challenges, these spaces have been opened for them. The nice thing about the early career researchers, or the ECRs, is that in our organization, we give them a voice. And they're part of our constitution. They are elected members who are part of the executive, and they have a voice in any of the organizational engagements. So they play a very important role in growing the organization. One of the other ways in which we engage with the broader community, but within our niche areas, is through the special interest groups. The SIGs allow researchers to find a discipline, their discipline and niche, and together they grow with researchers that are more experienced. Where we have a SIG coordinator and we have an overall SIG coordinator. And these are often people who have been running the SIGs for over a few years, and they're growing their leadership. Many of the members have access to these special interest groups, and they are able to grow and develop their niche areas. Within these early career research groups and the SIGs, they engage with policy. So we work not only theoretically through presenting our work at conferences, but we also work on the ground. We take the work, our knowledge production, take it to the ground by engaging with policymakers, connecting with national and international collaborators, and growing the disciplinary knowledge and research of South Africa and abroad. We also develop cutting-edge scholarship through our membership leadership. So we expose especially those researchers that are on the margins of research innovation and have limited access. We annually, together with our conference, we have the Nelson Mandela Legacy Lecture, in which we invite a very prominent researcher in South Africa with a large body of knowledge over years to present at the Nelson Mandela Legacy Lecture. And here we're able to expose researchers to, for instance, the A-rated scientists and scholars. We're able to expose them to NRF chairs, but we're also able to expose them to niche areas where it's growing, there's a body of knowledge, and more experienced researchers and scholars to talk about these niche areas and how it grew over time. We have our recognition awards, where doctoral master's honor students are recognized for their for the cutting-edge research within the institutions, where they wrote very prominent theses, etc., around a particular topic, and it was well written and it was well received by their peer community and examiners. We recognize people who are able to, who have grown their research over time. Sometimes there are those people within institutions that are often not given the platforms to be recognized. And we accept nominations. It goes through a very rigorous process, and we're able to recognize those people and expose young researchers to them. And we honor people who have been in the research arena for a long period of time, and we give them the accolades and the credits and the platform to be recognized. I think also, as I said, through our ECRs and SIGs, we allow access to research innovations. But what we're doing this year, and something quite different to previous years, is we are linking with the South African Educational Research Regional Network, in which we working with organizations outside of South Africa, and we've invited particularly African countries, where we are inviting them to be part of the conference and to play a role in the organization of the conference at the University of the Free State. So we are diversifying our research spaces. We're making it open and access to a larger group of people, where we can grow from one another, whether it's in South Africa or outside of South Africa. We also work with research institutions looking at connections abroad. Once again, the Journal of Education plays an important role in growing this recognition and also exposing the younger researchers to research innovation and more influential researchers in the area. So I think the final points that we want to emphasize as CSIR is that it's a very substantive research organization with a large research voice. And we're allowing young minds, young researchers, this access and exposure, this platform. CSIR has quite a solid reputation, and we attract young researchers across the board, people coming into the space with their more experienced supervisors, some higher education institutions bring in a large group of members every year in October to CSIR, and that's where we network and meet each other. The ECR plays a very important role in our strategic growth of CSIR. The ECRs have a voice, and their voice are very important for the growth and direction of the organization. CSIR also, we offer opportunities for mentoring support. While it is a challenge, the access to the more experienced mentors because of the time issues, we do offer these spaces because we make it part of our strategic planning and objectives to open up spaces for these opportunities. The special interest group plays a very, very important role in accessing research knowledge, particularly disciplinary knowledge, and taking this knowledge into the field, into our presentations at CSIR, into our ECRs and the SIG niche areas, into our Journal of Education. And this is where we're able to develop this access to this cutting-edge scholarship. We do have funding available for our early career researchers, but very limited funding, and it would be a good opportunity for these early career researchers in particular, and sometimes general members of CSIR, to access more funding. And we hope that any organizations that would be available to offer funding for students to come to our conference, because it's a good opportunity for them to be exposed and access the educational engagements in our community. And there's also a very robust publication platform, which we allow students and members to publish in accredited journals. So the take-home message is, CSIR is a safe space. We've worked over a long period of time to develop researchers within a community, a very active community. Our conference allows networking and collaboration, and CSIR gives opportunities to expose cutting-edge research and recognition. And I'd just like to thank you for allowing me to present today. We do have a conference at CSIR this year on the 29th of October to the 1st of November. So you're welcome to go to the CSIR website in order to have a look at other activities in our organization. So thank you. Thank you very much. Thanks, Yancy.
Fantastic. Thank you very much, Prof. Lakay. It's very good to hear that that education researchers have this home that they can where they can interact with colleagues and grow and take up leadership positions as well. Thank you very much for your presentation. There is a question in the chat to you from Mark Gordon. Do you have a niche area that focuses on the TVET engineering sector?
Yes, we do have a niche area, and you may go to the our website, and you can connect with the coordinator who would be able to give you more information. But we do have a TVET web niche area.
Okay, wonderful. And your website is what?
www.CSIR.co.za.
.co.za. Okay, all right. We'll distribute that also. Okay, that is good to hear. Do you have, while we're there, do you have a STEM education focus? Do you have a niche for that?
We do. We've got a curriculum niche area, and they take on a variety of engagements, and part of that is STEM. I come from a STEM background.
Oh.
And part of our curriculum, their focus is STEM, but also the TVETs. There's a focus on STEM as well.
Okay, that is very good to hear. Thank you very much,
Yes. Thank you.
That's good news.
Thanks.
I'll go to some of the other questions now, and we see how much we can fit in before 2 o'clock. Ronald Rachwasha says climate change-induced pandemics are evidenced through COVID-19 are a glaring reality. What work is being done on pandemic prevention and preparedness? I don't know if this is a question for patients? No, for Dr. Kokong, I assume. Dr. Kokong, would you like to answer that?
Thank you very much. The NICD is actually actively working on pandemic preparedness. Obviously, there's lessons learned, and I cannot go into specifics, but questions can be sent to us, and then we will respond to it. Thank you.
Okay, fantastic. Thank you. Yes, I want to mention just that it is so important and impressive that we've got a National Health Laboratory Service, which includes the NICD. We saw what an enormous role it played during the pandemic, and we have to be ready for the next one. We're all sick and tired of hearing of COVID, but we will have another pandemic. It's almost certain. So I'm very glad that we have this this safety net and this sense of preparedness. Zarina says, thank you to all the presenters on providing such meaningful insight into R&D in their respective fields and South Africa as a whole. Amanda Chulayu says, thank you, Dr. Malope, for the informative presentation. I'm interested in understanding the type of research that suffice grant approval. Which areas must the principal researcher focus on? Is there a possibility for animal-related research to be considered, especially if it focuses on biotechnology, nanotechnology, food security, consumer health, and policy development? Dr. Malope.
Yes, thank you very much, Yancy. Animal research will be supported as long as it's clear how it will contribute to improving pathology and clinical interventions. Nanotechnology, specifically NIOH, actually has a special unit on that. So we will specifically link them to the unit. So for every research that comes, as long as you have clear objectives and how clearly it will improve diagnostics or clinical interventions, we will try to review and link you to relevant people to work with you. Thank you.
Okay, fantastic.
Just to say that support obviously will come to researchers who collaborate if it's funding support with NHLS. But for the NHLS Research Trust, grant funding of 500,000, as long as you are in a pathology department, you will be funded. Thank you.
That is good news. Right, so they must go to the website. Will they be able to find the information there, or should they contact you?
I think our website is not up for now, but they can contact me directly. And okay. I think they can get my contact numbers from Ashley or any one of you, but they can contact me.
Yes.
And I don't mind to share my email address.
Fantastic, thank you. Ashley, if you're listening, or Mankwe, please post the email address in the chat. Okay. Dr. Merida Smith says, the question from the participant from Limpopo inquiring where to send an indigenous plant for analysis refers. The NSDF Forum is looking at the challenges of growing the S&I sector in South Africa. Aside from Dr. Perry, the emphasis has been on the formal scientific institutions in SA. The RDI investment survey would have been interesting if it had shown data from in 1994 and not only from 2000. That information is available on the web. I just want to inform you. Do have a look. Because there was a large exodus of scientists out of the government scientific institutions in that time. I'm assuming 94. Where did they go? They either went to existing technology companies or they started their own. As a previous senior researcher at the ARC, I left and started my own company in rural development, concentrating on entrepreneurial development in a deep rural area, KZN and Eastern Cape, assisting rural youth, women and people living with disabilities, developing small manufacturing companies and innovative products using primarily indigenous resources. You're overlooking an existing private scientist resource. Well, if we had known about this, Dr. Smits, we could have had a presentation on that. Do keep in touch. Let's see if we can't give you some airtime, maybe through our e-newsletter. But it is indeed a very interesting sector and difficult to measure and track. So the national statistics tend to focus on the formal sector, but you will remember that there was an aspect that also looked at NGO research and business research, and it didn't exclude small companies. So I think if they know about you, they can include you. Busisiwe said, if possible, can Prof. Mthunzi, is that Mthunzi probably, elaborate a bit on the molecular diagnostics of HIV/TB presence, please? I found the idea of putting the lab on a smartphone a very much needed and urgent innovation. Prof. Mthunzi, have you answered that question? I don't see Prof. Mthunzi.
Yancy, I think I saw her apologizing, saying she has to go to another Zoom meeting.
Oh, okay. All right. Then Mark Gordon says, greetings, colleagues, hoping that all have read the book, Why Innovations Live or Die, South Africa's Innovation System by the Mapungubwe Institute for Strategic Reflection. That looks interesting, indeed. Okay. I think that is it. Please tell me if I've missed something, and I open the floor for audience questions. Please put up your virtual hand. Okay, if not, I will go to the answered questions. Starting at the end. There is Sean Perry's email address. And Gordon Branson asked, my question relates to the how and why of integration of sustainability into the life cycle of the products presented. Oh, yes, I have read that one. And someone asked, Dr. Mchunu, it's a very interesting initiative about open science. Asked whether the Africa Archive is only limited to health science topics or diverse topics. And she said, no, it's a general repository. Okay, I think we have dealt with the questions. If you want to contact any of the speakers, please contact our office and ask us, and we can forward it to them. The presenters' presentations will be on the website, and usually they provide their email address on the presentations. So please remember, we will keep you updated through the NSTF e-news. So when the presentations are up, when the media release comes out, etc. Dr. Merida. Merida Smits. There, I've allowed you to talk. You may unmute yourself and talk.
Thank you, ma'am. Thank you for that answer that you gave just now. I don't mean that I want a platform or a forum. What I'm seeing a need for, because this is, as I've mentioned in my question just now, we concentrated yesterday and today very much on the national institutes, the research councils, the universities, and so on. And I'm a good researcher. I've won many awards in my capacity at the ARC and thereafter. And I concentrate on the needs of South Africa. And many of the mid-career, I mean, rocket scientists, all of us, who left between 94 and 2000, and it was because of a reason mentioned many times yesterday and today. We're talking about the problem of technology in South Africa and innovation, and we left because of bureaucracy. Yeah. Almost like Dr. Perry was saying, things weren't going fast enough. That's why he left. So I'm similar to him, and I know there are many like us who have left the ARC, the CSIR, HSRC, and my what I said to myself was, there's too much to do to write another report, to waste time writing another report. And I left. And my company's been going for 22 years. So good business person, good scientist, doing the stuff that our government and our president asks us to do every year in the State of the Nation address. What are our priorities? That you have to, if you're in the private sector, I say in inverted commas because it's small, you have to do what the country needs, otherwise you don't get funding. But what I'm seeing is, and by the way, my company is in the R&D survey every year. Lovely young man called Sintu who irritates me until I complete the survey every year. So I'm in all those sort of things, but I don't feel that there is a space for similar organizations. We are a resource. We're out in the field. We're at the coal face, so to speak. But let me try and write an article. Then I've got those publication costs that the lady mentioned. If I try and write an article, I've got to have an ORCID number? How do I get an ORCID number? I'm myself or my company. I always have to collaborate with universities and so on. And most of my staff are all agricultural graduates, highly qualified, and always wanting to do the next MSc, PhD, and so on. But we have approached universities twice and wanted to insist that the intellectual property of the research we wanted to do was ours, because we have got a couple of patents and trademarks, because we do product development, and then the universities would not be willing to sign an IP agreement. So I'm saying that there are policy issues and there's discussion needed around creating a home, if you like, but let's not bureaucratize it, but that we can apply for national grants because of the work we're doing. Just because we're not at a formal research institution, we are doing cutting edge stuff with our own funding or from overseas donors, or I get a lot of my money from BEE, corporates looking to get their BE score in enterprise development. So I'm being innovative in my funding model, for example. But the work we're doing, I'd love to publish it. And I've written it all up, but where to publish and pay the fees and under what banner am I writing it? You see what I'm saying?
Yes. Yes.
Okay, that's my bit.
Thank you very much, Dr. Smith. So it's good to hear from you, to hear your voice, and to understand what these issues are. And those are very concerning indeed.
Just add that to the mix.
Yes, it seems like it is worse than than being at a formal institution. I mean, all the challenges actually just become exponentially bigger.
Yeah, the innovation is there, but the platform to encourage that innovation is not there.
Right. What is your company's name?
Scientific Roots. So it's scientific and then Roots, ROOTS. The incubator, AgriBiotech, the place where we do the product development and entrepreneurial development, is called AgriBiotech, TECH. And that's a NPC. So we've got the PTY, which is Scientific Roots, which is accredited for skills development and so on. And then AgriBiotech is the incubator. And we are developing some amazing entrepreneurs, very much in the Dr. Perry realm. Innovative use of indigenous resources. And we talk about innovation and skills development and scientific graduates. I'm working with rural youth. These are school leavers, rural women, middle-aged women, people living with disabilities, and with the right mentorship and what I call targeted creativity, the products they're coming up with blow your mind. And then my job as the scientist in the bunch is to do the efficacy, shelf life, and safety research.
My
You
goodness.
see how that fits, you see how that fits together. The
Wonderful.
innovation is not just scientists. It's in the man who makes the boerewors, or Oma Rusks.
Yes.
Those are the stories I like to use. But they're coming up with novel beverages, health food products, all based on speaking to their Oma or speaking to their auntie.
Yeah.
What did you use to fix that cow, Oma, last time? Then they come and we look at the plant and then my research kicks in.
Right.
So they have this novel little idea, but then when I start packing it with the research side of things, I mean, that's innovation. And that's what our country has been asking for.
Indeed.
But we rely only on the scientists. I gave the keynote address at the 2012 Women in Science Awards. Okay. And if anybody was in that audience, I spoke about, and I don't mean any disrespect, but the science and innovation structure in South Africa pays for scientists to sit behind their desks and chew their pens. And I use that as an analogy of blue sky research. They sit there and they come up with stuff. We need blue sky research. But in South Africa, there's too many problems and too many solutions just lying out there.
Right. Yeah, so this sounds like grassroots innovation.
Very much.
You call it that.
But it needs research to verify it.
Yes, indeed.
And then I can put that verification. Everything's done, GLP, good laboratory practice, all these international standards that I use in the research. And I can write a pamphlet that goes in the product box, giving the results. But wouldn't it be nicer if that I can just put a reference to an article?
Yes. No, indeed. It's a huge challenge. But Tia has been working on grassroots research and entrepreneurship. Have you been in touch with them lately? I think it's fairly recent.
I went to Tia, I would say 7 to 10 years ago. but I wasn't pleased with the intellectual property rights processes. And because the patent that we have subsequently done was extremely novel, and I just felt there were too many people were touching my papers. You know what I mean? I just felt that there were too many people.
It
And
might still not be it might still not be suitable, but the Department of Science and Innovation is having a drive where they're working with grassroots innovators, so it might be worth just checking it out.
Okay, I'll do that.
Yeah, fairly recent development. The past
Is
2
it that
3 years.
is it that Sesame Fund?
I don't know what it's called.
Innovation Bridge, because I wrote them all down yesterday. Innovation Bridge portal. Might be one.
No, it's look, it might be included in the Innovation Bridge portal, but I specifically know that they are pushing that from a policy point of view, and they have some awards as well at the SFSA for grassroots innovators.
Okay, I'll have a look. Thanks, man.
Okay, all right. Thank you very much, Merida. Tiamu.
Thank you very much, Yanzi. Apologies, I had to leave earlier because we had a South African forum, South Africa Science Forum committee meeting.
Okay.
And I put an announcement on the chat because I do sit in that committee. As you know, the forum seeks to really ignite conversations. I think this year's theme is igniting conversations about science with the sub-theme on innovation and science for humanity.
Good.
So I think stakeholders and organizations are really urged to think about submitting proposals for side events. I think you'll see there on the website, the major tracks really around youth in science, technology and innovation, STI, and Africa. Science, technology and innovation partnerships in the G20. You'll recall that South Africa is going to be leading the G20 presidency in 2025. So there's an opportunity there to also engage at that level. There's also a track on bridging science and economic diplomacy in policymaking. There's a track on shaping innovation, powering the future industries. And then there's a broader theme on global science for humanity, really covering the areas of agriculture, health, climate change, and just transition. And then the very important issue of bringing science communication and science journalism together. So I wanted just to pass on the message from the committee that stakeholders are urged to really provide proposals. I think the deadline is 23rd of August. I don't know if it will be, if it will move back. The area again for the themes is around side events, exhibitions. There's also an opportunity for Science Diplomacy Award nominations. And of course, the general themes that has just been outlined in the list that I provided. That's one thing, Yanzi. And then the other thing that I really wanted to emphasize also in this session is that there is the African dimension to the STI from the South African perspective and other countries. There is an opportunity through the current revision of the Science, Technology, Innovation Strategy for Africa, STISA 2024. As you know, this strategy is elapsing 2024. So there is a process to do a review, which has been conducted and completed. Now there's a process to develop the 2034 strategy. There's a draft. I think there's still a window of opportunity to provide inputs so that this 10-year strategy could be shaped, including by inputs, some of which you discussed in this particular sessions, Yancy. Very good inputs by stakeholders. And I think you can coordinate with DSI through MAMPE to provide those inputs because I think they are relevant to STISA 2034. There's also a report that is out that was conducted by Professor Mugabe, who is working with the AU on the review of STISA 2024. Very good findings, some of which overlap with what you said. I can also share that report because I think the discussions are related. So I thought I'd mention those two elements to your stakeholders here. Thank you.
Thank you very much, Tiamu. Indeed, that's very useful. So that is the SFSA, and have a look on the DSI website for that, but we'll also put it in the NSDFE news, and the revision of the SDI strategy for Africa and there might be a window of opportunity for inputs, but I will take up contact with the department, with
And
Mampe.
incidentally, also the Africa AU AI strategy has been developed and finalized to an extent. There's a window for stakeholder consultations regarding the implementation plan. Again, these things move very quickly. I think they could be consulted better. But the train is moving. Everybody's been talking about data, AI applications in these sessions. So again, there's an opportunity through the AI strategy for the continent to be able to influence, I think, the implementation plan and probably aspects of some of the activities. So I think UNESCO has been assisting with that. So hopefully there will be a formal announcement to that end. We attended the Department of International Relations and Cooperation organized session for G20 visioning for South Africa. And I think an announcement that was made by UNESCO that the AI strategy for Africa will be announced soon. So that's another element to look out for, given that this strategy is in a way a continental conduit for collaborations. So I would imagine in the implementation plans, there must be avenues for your organizations here to be able to engage in the continent. Thank you.
Indeed, that would be very useful. What is the deadline for that? Do you know?
I think for this STISA 2034 input, check with DSI. Mampe is the contact. She's collating the inputs for STISA. In fact, there was a workshop organized by DSI for stakeholders to engage. I don't know if there'll be a second round of consultations, but I think the window is very tight. Do check with her if you can still provide inputs.
All right.
Okay,
Will do.
Thank you.
Thank you very much, Tsiamo. That is a great note on which to end. Let me just check. There is a question. Well, there's the information from Tsiamo. And there is, let's see.
That was the one on SFA-SA.
Yes.
I just pasted the links and the information on the chat.
That's right. Yeah. And then Mkhize says, good day all. I just want to appreciate the valuable information that was shared with us in the past 2 days. Of concern to me is the fact that, as Dr. Mustafa explained, government is the biggest funder of research. We're going to see a decline in research that is funded by government because the government is quite low in funds. We see budget cuts in the government departments. So it looks like the other role players in the research fraternity, especially from the private sector, will have to support more research as long as it is for the good of the country's development. Apologies for a long message. Not at all, Mkhize. That's you're very welcome. And it's a very important point to make. I also think we must keep urging government to increase research. The indications are that they will, but keep up the pressure. Busisiwe says, great to have education being represented in this forum. Thank you. With the pressure on HEIs to serve society in new ways, what innovation is CSIR bringing to the table in this regard, particularly the STEAM group? Dr. Lucky?
Yes. So in our conference, we have a large attendance of, we have a STEM group. We've got a large attendance in our STEM group. And also we've got great connections with STEM and with STEAM as well, because a lot of our science education people are involved in STEAM. So it's growing within our organization. We don't yet have a particular niche area related to STEAM, although it's under the curriculum and TVET, as I said earlier. But this is growing, and I do have a science education background, like I said, and we are trying to push the development of STEAM within CSIR as well. Thank you.
Fantastic. Thank you very much. Ken Concepts says, hi, is there anywhere I can help in technology, AI, rapid prototyping, and 3D designing? I would love to take part. Anyone who wants to answer that question? Okay. No, I don't think so. It is now 2:01, and I think we have run out of steam and STEM and everything out of discussion forum for today. I want to thank everyone who attended, everyone who has stayed up to this point. There are 98 of you. Thank you very much for attending, giving your input, listening, and for what you are going to tell other people that you learned here today, and the kind of issues that are very important for us to take up and go forward. To our presenters, a special thank you to you. It has been such a rich 2 days, so much information to take in, and the insights also regarding what the priorities are and the kind of areas that people haven't been aware of that should be prioritized. Going forward, like I said, we will be having a committee meeting with the Science Council's representatives to discuss how to take the outcomes of this discussion forum forward. The presentations will be on the website, the video recordings, and we will be having a media release with summarized issues, as well as recommendations. So have watched the space being the NSTFE newsletter, and do check your spam box so that you don't lose the information. With that, I declare this discussion forum closed, and I wish you a great rest of the day and the week. Goodbye.
Bye. Thank you, Yancy.
Bye. Thank you, Vilna. Thank you, Mankwe. Thank you, Jane.
All thanks to you, Yancy, for hosting the event. Thank you.
Thanks for your postings in the chat.
Bye.
Bye.
Bye-bye.